Hi everyone, welcome to all. This is Vidyam Academy's NEET Initiative Most Expected Question Series. This is Rajiv Patel. Today's topic is 12th Biology, second chapter, Human Reproduction. Into this subtopic is Gametogenesis, Menstrual Cycle, Fertilization and Implantation. Let's welcome to all. Let's get started. The first question is, in the human male, spermatogenesis occurs in the seminiferous tubules. Which cell types provides nutritional support to the developing sperms? And which cell type secretes testosterone? Option A, Sertoli cells, nutrition, Leydig cells, testosterone. Option B, Leydig cells, nutrition, Sertoli cells, testosterone. Option C, Sertoli cells, nutrition, spermatogonia, testosterone. Option D, Sertoli cells, testosterone, Leydig cells and nutrition. Would you guess the answer? What is the exact answer for this question? The correct answer is option A. Sertoli cells, nutrition, Leydig cells, testosterone. Why and how? Let's see the explanation. Inside the seminiferous tubule, Sertoli cells line the wall and extended inward, physically cradling the developing sperms at every stage from spermatogonia through spermatozoa. They supply nutrients, growth factors and hormonal signals that keep the developing cells alive and progressing. They are sometimes called nurse cells of the testes. Leydig cells, also called interstitial cells, sit in the connective tissue between the seminiferous tubules, completely outside the tubule wall. Under the stimulation by LH from the pituitary, Leydig cells synthesize and secrete testosterone, the male sex hormone that drives spermatogenesis, secondary sexual characters and libido. So these two cell types have completely distinct location and roles, and swapping them is the most exact answer and explanation for this question. And next, move to question number 2. A primary oocyte is arrested at a specific stage of meiosis and remains arrested for years. At which stage is it arrested and which stage does the secondary oocyte arrest? So here the options are. Option A, primary oocyte arrested at metaphase 1, secondary oocyte arrested at metaphase 2. Option B, primary oocyte arrested at prophase 1, secondary oocyte arrested at metaphase 2. Option C, primary oocyte arrested at prophase 2, secondary oocyte arrested at metaphase 1. Option D, both primary and secondary oocytes are arrested at prophase 1. Would you guess the right answer? Your time starts now. The answer is option B. Why and how? Let's see the explanation. The primary oocyte is formed from the oocyte during fetal development. It enters meiosis 1 but gets stuck very early at prophase 1, specifically at a substage called dictotin, also called the diplotin arrest. This arrest is maintained by high levels of cytoplasmic factor and can last anyway from 12 to 50 years, the entire span from fetal life to the monthly ovulation cycles. Each month, the LH surge forces one primary oocyte to complete meiosis 1, producing the secondary oocyte and the first polar body, so both haploid. The secondary oocyte immediately enters meiosis 2, but arrests again at metaphase 2 and stays frozen here until a sperm fertilizes it. Fertilization triggers, it releases the inside the oocyte, which releases that metaphase 2 arrest and allows meiosis 2 to complete. So this is the right and correct explanation to the question of 2. Let's move to question number 3. During which phase of the menstrual cycle does the endometrium proliferate and thicken, and which hormone primarily driven this change? Option A, luteal phase driven by progesterone. Option B, menstrual phase driven by LH. Option C, follicular phase driven by estrogen. So here follicular proliferative. Yes, option D, ovulatory phase driven by FSH. What is the answer? The answer is option C. Follicular proliferative phase driven by estrogen. So this is the right answer. This is the explanation. Let's see. After menstruation strips the endometrium bare, the follicular phase begins around day 6. During this phase, the growing ovarian follicle secretes rising amount of estrogen under FSH stimulation. Estrogen acts directly on the endometrial cells, causing them to proliferate. They multiply. The endometrial thickness increases from about 1 mm to 10 to 12 mm. New blood vessels grow in and uterine glands lengthen. So this is why the phase is also called the proliferative phase. The endometrium is rebuilt and thickened in preparation for a possible embryo. After ovulation, progesterone takes over during the luteal phase and switches the endometrium from proliferative mode to secretory mode. The glands start producing nutrients. Estrogen builds the lining, progesterone prepares it for the embryo. So this is the explanation why follicular phase or proliferative phase driven by estrogen is exactly answer for this question. Next, move to question number 4. When a capacitated sperm contacts the zona pellucida of the secondary oocyte, a specific reaction occurs that allows the sperm to penetrate the zona. What is the reaction and what does it involve? So here the option A, cortical reaction. Cortical granules from the oocyte releases enzymes to block polyspermy. Option B, acrosome reaction. The acrosome of the sperm releases hydrolytic enzymes that digest the zona pellucida. Option C, capacitation reaction. Sperm gains the ability to swim faster inside the zona pellucida. Option D, zona reaction. The zona pellucida softens to allow sperm entry without enzyme action. Would you guess the right answer? Yes, the answer is B. Agrosome reaction. The agrosome, the sperm releases hydrolytic enzymes that digest the zona pellucida. Why and how? Let's see the explanation. The acrosome is a cap-like membrane bound structure at the tip of the spermid formed from the Golgi apparatus during spermatogenesis, and it's packed with the hydrolytic enzymes including acrosin. When a capacitated sperm binds to specific glycoprotein receptors on the zona pellucida, this binding triggers exotosis of the acrosome. The acrosomal membrane fuses with the sperm's plasma membrane and the enzymes are released directly on the zona pellucida surface. These enzymes chemically digest a narrow pathway through the zona, allowing the sperm head to the tunnel through and reach the oocyte plasma membrane. This entire process is called the agrosome reaction, and it is the essential molecular step that converts zona binding into actual sperm penetration. Without it, even a capacitated sperm cannot fertilize the egg. This is the right explanation what the question number 4 demands for. Let's move to question number 5. Which stage of the embryo plants into the uterus and in which specific uterine layer does implantation occur? Option A, mola implants into the myometrium. Option B, blastocyte implants into the myometrium. And option C, blastocyte implants into the endometrium. Option D, gastrula implants into the endometrium. So here the options A, B, C, D. Did you guess the right answer? Yes, option C is right answer. Why and how? Let's see. After fertilization in the ampulla, the zygote undergoes cleavage while traveling through the fallopian tube, producing a solid 16-cell morula. As the morula enters the uterine cavity, it absorbs fluid and reorganizes into the blastocyte, a hollow sphere with an outer trophoblast layer and an inner cell's mass. Before implantation, the blastocyte sheds the zona pellucida in a process called zona hatching, which exposes a trophoblast cells. These trophoblast cells then make direct contact with the endometrium, the innermost granular lining of the uterus, and begin secreting enzymes that digest the endometrial cells, allowing the blastocyte to burrow in. This occurs approximately 6 to 7 days after fertilization. The myometrium is the thick muscle layer beneath the endometrium and is never the site of normal implantation. That's what. So this is the right explanation. And next, move to question number 6. Which of the following correctly represents the complete ploidy sequences in spermatogenesis from spermatogonium to spermatozoa? Option A, spermatogonium, 2 nucleus, primary spermatozoa 2n, secondary spermatocyte n, and spermatid n, spermatozoan n. Option B, spermatogonium, primary spermatocyte 2n, secondary spermatocyte 2n, and spermatid n, spermatozoan. Option C, spermatogonium 2n, primary spermatocyte 2n, secondary spermatocyte 2n, spermatid n, and spermatozoan n. Option D, spermatogonium 2n, primary spermatocyte n, secondary spermatocyte n, spermatid n, spermatozoan n. What is the right answer? Yes, the answer is option A. Why and how? Let's see the explanation. Spermatogonia normally diploid 2n body cells lining this seminiferous tubule. Some spermatogonia enlarge and differentiate into primary spermatocytes still diploid 2n because they have not yet divided by meiosis. Each primary spermatocyte then undergoes meiosis 1, which halves the chromosome number, producing 2 secondary spermatocytes, each haploid n. This is where ploidy drops for the first time. Each secondary spermatocyte that undergoes meiosis 2 and it produces 2 spermatids and still haploid n because meiosis 2 doesn't change ploidy. Finally, the round spermatids undergo spermatogenesis, a morphological transformation involving no division, to become elongated motile spermatozoa, still haploid. So one primary spermatocyte ultimately produces the 4 haploid spermatozoa. So this is the answer for the 6th question. Let's move to question number 7. Consider the following statement about hormonal events in the menstrual cycle. Statement 1. Rising estrogen from the maturing follicle events experts positive feedback on the pituitary triggering the LH surge that causes ovulation. It's a statement 1. Statement 2. After ovulation, the corpus luteum secretes progesterone and some estrogen, which together exert negative feedback on the pituitary to suppress FSH and LH. Statement 3. If fertilization does not occur, rising progesterone in the luteal phase causes the endometrium to shed, triggering menstruation. So here statement 1, 2, 3 we have. Consider the following statements about hormonal events in the menstrual cycle. So here the options are: option A, all 3 statements are correct. Option B, only statement 1 and 2 are correct. Option C, only statements 1 and 3 are correct. Option D, only statement 2 is correct. What is the answer? It's very, very easy question. Would you guess the answer? What is the answer? Option B. 1 and 2 statements are correct. Why and how? Why other 3 is wrong? Let's see the explanation. Statement 1 is right because estrogen initially exerts negative feedback on the pituitary at low levels, but once it rises above a critical threshold near the end of the follicular phase, the feedback switches to positive. This high estrogen level stimulates the pituitary to release a massive surge of LH and a smaller surge of FSH, which triggers ovulation of the Graafian follicle. Statement 2 is also correct because after ovulation, the corpus luteum secretes both progesterone and estrogen. Together, they feed back negatively on the hypothalamus, pituitary, suppressing GnRHs and FSH and LH. This prevents any new follicle from developing during that cycle. Statement 3 is wrong because it is for the progesterone, not its rise, that triggers menstruation. When the corpus luteum degenerates, progesterone drops, causing the endometrial blood vessels to consist and the endometrium sheds. That's what that third one statement is wrong and 1 and 2 are right. Next, move to question number 8. So here match the following column 1 and column 2. So here match the following, one side we have terms and another side is a correct definition. So option 1 here, spermatogenesis. So here the correct definition is process by which sperms acquires fertilizing ability in the female tract. Option 2 here, spermiation. So here option B is morphological transformation of spermatid into a mature spermatozoan. So here option 3, capacitation. So here option C, release of mature spermatozoa from Sertoli cells into the table lumen. So here option 4, ovogenesis. Option D, formation of mature female gametes from ovozonia. So here the options are option A, 1B, 2C, 3A, 4D. Option B, 1A, 2B, 3C, 4D. Option C, 1C, 2B, 3A, 4D. Option D, 1B, 2A, 3D, 4C. What is the right answer? Would you guess the right answer? Option A, 1B, 2C, 3A and 4D are correctly matched, and which explain term and correct definition. Let's see why and how. Spermatogenesis is the transformation process around non-motile spermatid undergoes dramatic shape changes including nuclear condensation, acrosome formation from the Golgi apparatus, a flagellum development from a centrole, a development from a shedding of excess cytoplasma to become a streamlined motile spermatozoon. This is morphological transformation, matching B. Spermiation is the specific event of releases after spermatogenesis is complete. The finished spermatozoa are physically detached from the Sertoli cells. Spermiation is to the specific event of release after a spermiogenesis is complete. The finished spermatozoa are physically detached from the Sertoli cells and released into the fluid filled lumen of the seminiferous tubule, matching C. Capacitation is the physiological priming process that freshly ejaculated sperms undergo inside the female reproductive tract. Changes to the sperm membrane increase motility and ability to undergo acrosome reaction, without which fertilization cannot occur, matching A. Oogenesis is simply the complete process of a egg formation from the ovogonia through to the mature ovum, matching D. And question number 9. Assertion A and we have reason R. Assertion is after the first sperm successfully fertilizes the secondary ovocyte, additional sperms are prevented from entering. This ensures only one sperm fertilizes each egg. Reason R. Fertilization by the first sperm triggers the Cortoli reaction. Cortical granules beneath the ovocyte membrane fuse with the plasma membrane and release enzymes into the pervitoline, a space which harden and chemically modify the zona pellucida, making it impenetrable to further sperms. Option A, both A and R are true and R is not the correct explanation of A. And option B, both A and R are true but R is not the correct explanation of A. And A is true but R is false. And option D, A is false. Multiple sperms routinely fertilize the same egg. What is the right answer? Option A. Both A and R are true and R is the correct explanation of A. Why? Polyspermy, more than one sperm fertilizing the same egg must be actively prevented because it would result in an embryo with an abnormal number of chromosomes, which is almost always lethal. This prevention is not passive, it requires an active cellular response. Reason is R also true because it gives the exact molecular mechanism. When the first sperm fuses with the ovocyte plasma membrane, it triggers a wave of calcium ions that sweeps across the ovocyte. This calcium wave causes thousands of cortical granules, small vessels stored just beneath the ovocyte membrane, to undergo exocytes, releasing their enzymes content into the pervitalized space between the ovocyte membrane and the zona pellucida. That's what option both A and R are true and R is the correct explanation of A. And question number 10, complete sequence. Arrange the following events in the correct chronological order from spermatogonium formation to blastocyte implantation. Arrange the following events in the correct chronological order from spermatogonium formation to blastocyte implantation. PQRSTU. Acrosome reaction. P. Q. Blastocyte sheds zona pellucida. Or LH surge ovulation. Yes, secondary spermatocyte undergoes meiosis II. T. Capacitation of sperm in a female tract. Option U. Trophoblast invades endometrium. So here the options are A. SRTPQU, B. RSPTQU, C. STRPQU, and D. RTSQPU. So what is the right answer? And arrange the following events in the correct chronological order from spermatogonium formation to the blastocyte implantation. So here the right answer is option A, SRTPQU. Because the secondary spermatocyte undergoes meiosis II during spermatogenesis in the testes, producing spermatids that mature into spermatozoa. This happens continuously in males before any particular fertilization event. R comes next. The LH surge triggers ovulation on day 14, releasing the secondary oocyte into the fallopian tube. This is the event that makes an egg available for fertilization. T follows because sperms deposited in the female tract undergo capacitation inside the uterus and fallopian tube, acquiring hypermotility and the ability to perform the acrossing reaction. This must happen before they can fertilize the egg. A capacitated sperm contacts the zona pellucida of the secondary oocyte in the ampulla, triggering the acrosome reaction, releasing hydrolytic enzymes that drill through the zona and allow the sperm to reach the oocyte membrane. After fertilization, the zygote undergoes cleavage and develops into the blastocyte, which then sheds its zona pellucida to expose as trophoblasts. Finally, so U follows because the trophoblast cells of the hatched blastocytes invade the endometrium, completing implantation. That's what. So these are the sequence which is correctly and properly arranged. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.